This figure illustrates the different synapses between neurons. So in this case, we call it axodendritic synapse, which is the most common type of synapse. And then comes the axosomatic synapse between the axon terminals and the cell body of the neuron. And the least common is the axonic synapse, which is between the axon of the presynaptic neuron and the axon in the postsynaptic neuron, or what we call it mostly a presynaptic inhibition, that we will talk about it later. Now this figure is showing a neuron, a typical neuron, that we will divide it into 3 zones. This zone from here to here, that includes the dendrites and the soma, we call it the input zone, because most of the input, the information coming to this neuron, will come through the dendrites, then the cell bodies or the soma. The next one, or the next zone, is the integrative zone. And here is at the axon hillock, where the integration of these inputs will be processed here, and to decide whether to proceed as an action potential throughout the axon or not, through this conductive zone, which will conduct this type of information to the axon terminals, in which they will synapse with the next neuron or neurons. Now, different functions in these different parts of a neurons may be result from the different distribution of ion channels. As you noticed here, the blue color is a ligand-gated channels, whereas the red color is a voltage-gated channels. And you know previously that in order for action potential to take place, we should have a voltage-gated channels. And you can see that the distribution of these channels is mainly at the axon hillock, and then at the nodes of Ranvier and the axon terminals. And there is the conductive zone, the integrative, and the conductive zone where the action potential takes place. However, in the soma and the dendrite, it's mainly ligand-gated channels. And so therefore, what happens here, the change in the membrane potential, we call it postsynaptic potential, and here is action potential. Therefore, it's important to know that the generation of action potential does not take place at the site of stimulation anywhere here, but actually it's on the axon hillock because of the highest percentage of the voltage gated channels, and so therefore has the lowest threshold to reach to action potential. And as you know previously that each type of cell has its own resting membrane potential, thus any change toward the positive charge, less negative number, will be called depolarization, results in excitation because it's reaching toward the threshold, whereas any change to more negative number, hyperpolarization, will be inhibition of this neuron because it will be further away from the threshold. This is the resting membrane potential of this neuron, which is minus 65. Here it comes an excitatory signal, which results in influx of sodium ions. So more positive inside, less negative number. It's minus 45 compared to minus 65. That will result in reaching, if it reaches threshold, then there will be activation of the and spread of action potential. On the other hand, an inhibitory signal, either by increasing the influx of chloride or the efflux of potassium, results in hyperpolarization. Now the membrane potential is minus 70. This neuron is inhibited, no action potential will be transmitted. Again, we said that action potential does not start near the signal, but it does start in the initial segment of the axon or the axon helix because of the higher concentration of voltage-gated sodium channels, and which means lower threshold at that area. You can see here that the dendrites are very long, of various lengths, and they are very branched, so that provides a large spatial field in these areas and a great opportunity for summation of signals from different types of presynaptic neurons. Now, because of the membranes of the dendrites are long, they are thin, they are more leaky to potassium and chloride, that will help in what we call it a decremental conduction. So a large share of the membrane potential will be lost as it reaches the soma or the cell body. Let's assume that this excitatory postsynaptic potential from neuron 1 is equal in magnitude to 2. Which one do you think will have a greater effect on this type of a neuron? Now let's assume the effect is either by the green neuron or the blue one. The green one has an excitatory postsynaptic potential on this neuron equals 10 millivolt, while this neuron has an inhibitory IPSP postsynaptic potential equals to minus 5 millivolt. What do you think will be the net result on this neuron? Now the soma or the cell body, as you see here, it has a large diameter, which means less resistance to conductance, in addition to the high conductive electrolytic fluid that's inside the soma, which means that any change in membrane potential will be transmitted equally to all parts of the soma. This is very important principle for the summation of signals from different neurons when they reach the soma, because one excitation or one EPSP, excitatory postsynaptic potential, is almost always never enough to excite this and reach a threshold which needs at least 10 to 20 millivolts to reach a threshold for excitation. For example, an EPSP of 5 millivolt will not result in reaching a threshold and firing an action potential. However, having an EPSP of 10 millivolt and another one of another 10 millivolt can be summated because both of them are excitatory and they will be transmitted equally through the soma membrane. So the summation will be 20 millivolt that can reach a threshold and activate these voltage gated channels and producing an action potential. And as you know previously, that summation can be either a spatial summation in which simultaneous activation by two widely spread signals can be summed into another one which can reach a threshold like this. This is called spatial summation. The other type is temporal summation, in which frequent stimulation, it can be from the same point, results in higher change in the membrane potential, higher amplitude, and may reach to threshold and fire and action potential. This graph is showing that the higher number of excitatory postsynaptic potentials, the more the chance of reaching to action potential. For example, this blue one, it's a summation of 4 synapses, whereas the green one is 8 excitatory synapses, where the red one is 16 excitatory synapses that are summated and then results in an action potential in that neuron. This graph showing a summation of 2 excitatory postsynaptic potentials and 1 inhibitory postsynaptic potential on the soma. So let's say if it's 10 EPSP, 5 EPSP, and 10 IPSP, what will be the net postsynaptic potential in this neuron? Is it gonna fire an action potential or not? This is another example showing there's so many synapses on this neuron, and these synapses can be excitatory, the E, or I, inhibitory. And you can see the decremental conduction in the dendrites, and then the summation at the soma, then whether this will fire an action potential or not in this type of a neuron. You can see here what's special, that there is inhibitory postsynaptic potentials affecting the axon hillock itself. So that results in hyperpolarization and no action potential firing in this neuron, despite the larger number of excitatory synapses on the dendrites here in this neuron. We talked before about the postsynaptic inhibition. There is another example of inhibition. We call it presynaptic inhibition. And here you can see an axonal synapse, in which before the main synapse between the presynaptic and the postsynaptic receptor, this axon will release an inhibitory neurotransmitter through these ionotropic receptors. So mainly it will be GABA activating chloride receptors that will result in hyperpolarization, preventing the action potential from reaching here and activating the exocytosis of the vesicles. That's why we call it inhibition, and it's presynaptic because it's before this main synapse that's occurring. This is an important feature in many sensory information or sensory receptors, such as the ones that are present in the eye, in order to have a better contrast of vision. They do what we call it presynaptic inhibition of the nearby receptors. Now the excitatory postsynaptic potentials can be there affecting the neuron, but not enough to reach an action potential, not enough to reach a threshold. Therefore, there is no action potential, but this neuron is at nearer state to reach this action potential. It's facilitated to receive another excitatory signals, so it may reach this threshold. That's why we call this process facilitation of a neuron, in which a summation of the postsynaptic potential is excitatory but not enough to reach a threshold yet. Now, when the summation is more toward an excitatory drive, we call this a neuron is in an excitatory state. On the other hand, if the summation is more toward inhibition, so we call it an inhibitory state. In both circumstances, they haven't reached threshold, but they will be in excitatory state, so it's more facilitated and near a threshold, but not there yet. This graph is showing that neurons not only are different in their excitatory state or the threshold at which they reach and then will start to fire an action potential, also they differ in the frequency of discharge of the action potential. For example, in neuron 1, it has the lowest threshold, so it can be activated easily and start firing action potential. But the maximum frequency of discharge is lower than the one for the blue neuron or the neuron 3, which has the highest threshold. So almost there is no relationship between the excitatory state and the frequency of discharge. It's related to the function that this neuron is supposed to do. Do you think this neuron will fire an action potential or not? If the resting membrane potential is minus 65, threshold is minus 55, it has an EPSP of 10, IPSP of 20, and EPSP of 5. What do you think? Thank you. Our last lecture will be next time, talking about neuronal pools and neuronal circuits.